Now we'll see the DNA sequencing, how it is performed and what are the problems in DNA sequencing. So we consider that we have a few magazines, journals, and we cut these journals into the millions of small pieces. And we cut in such a manner that one of the journals they cut into smaller sizes and other are relatively bigger sizes, so that the smaller sizes they overlap with the pieces of the journals that have some larger pieces. So in these pieces that we have done in for these journals, we consider that most of the pieces of some journals they are lost, and some of the pieces they were painted and we cannot read the text on these pieces. So same is the problem with DNA sequences. So the most of earlier DNA sequencing techniques were used, and they were only used to read the 500 to 700 nucleotides of one experiment. It means that we can read only 500 to 700 nucleotides at one time. And we can assume that this one fragment of 500 to 700 nucleotide, it can be compared with one of the pieces that we have done for the magazine. Then when we perform the DNA sequences and get the fragments, then the problem is again that we have to assemble those fragments into an entire genome. And the problem is again, as we encounter when we did the small pieces of the magazines, that some of the pieces they were lost. And in the DNA sequencing also, some of the sequence they never come up in the sequencing process. And sometimes we cannot read the sequence properly. And this problem is unavoidable because there are experimental errors when we do the DNA sequencing. So there are two sequencing methods that were put forward by different scientists. One of the method was developed by the Frederick Sanger, and he was the scientist working in the Cambridge University, United Kingdom. And the other scientist was Walter Gilbert, and he was working in the MIT institutions in the United States. So Sanger has the method developed by Sanger was much better as compared to the Gilbert method, because Sanger method was considered as the biological method. And in that method, he used to sequence the unknown DNA fragments by adding one of the nucleotides one by one. So when he was performing these experiments, he came up with the idea that he can produce the sequences of different sizes, and this will help in the sequencing of the DNA fragments. And by doing so, he will come up with the different sizes of the fragment, and then he can sequence all those fragments and find the actual sequence of the unknown fragment. As we have seen that the Sanger method was better than the Gilbert method, and Sanger discovered that he can produce the fragments of different sizes. For example, here we can see if this is the fragment size, fragment that is ACGTA, GCTA, then we can produce the different fragments of different sizes if we limit one of the nucleotides out of 4. In this case, we can see that we have limited the thiamine in the sequencing process. If thiamine is limited, then whenever there is a thiamine in the sequence of the unknown fragment, at the thiamine the fragment will be terminated. For example, here is the position of the thiamine, then what we will get is only A, C and G. So other possibility is that despite it is not terminated here, but it will be terminated at the second T, before second T, and the fragment that will be obtained is of this size. So another example is that the same nucleotide fragment, and what we will limiting is guanine, that is missing. So if guanine is missing, then whenever there is a guanine in the fragment, before guanine the process will be terminated, and what is obtained is two fragments. One is AC, and if there is a second possibility that guanine is here, then it will be terminated from here, and we will obtain ACGTAA. So by using this technique, we can see that fragments, different fragments, they are obtained. If T is limited, this fragment is obtained 3 nucleotides, and if T is again limited, we will obtain 1, 2, 3, 4, 5, 6, 7, 8 nucleotide fragments. And in the other case, if G is limited, then we will obtain this fragment of 2 nucleotide and again another fragment of 6 nucleotide. So we have 6, 9. So we can see, just limiting the 2 nucleotides, we have fragments of different sizes, 2, 3, 6 and 9. So if we limit the other 2 nucleotides, adenine and cytosine, then again we will find the nucleotides other than these fragments. So by using this technique, different fragment size they can be obtained, and each of the 4 starvation experiments produce a ladder of fragments of varying lengths, and these are called the Sanger ladder. And the later Sanger found chemicals, and these are called the dideoxynucleotides, that could be inserted in place of these adenine, thymine, guanine, adenine, cytosine, and cause the growing DNA chain to end. So by using these dideoxynucleotides, that was very easy, that if T is dideoxythymine, then whenever there is a T, the reaction is automatically terminated and will get the fragments of different sizes. And then we can sequence these small fragments, and we can determine the sequence of the original unknown fragment.